Great. So I'm really happy to introduce Tyler Korman. Tyler is a Northern California native who managed to find happiness in wonderful Southern California. He did his undergraduate work at UC San Diego, where he was also actually on the UCSD soccer team. So if you need a player for your soccer team, he'd be a really good guy to recruit. He moved to UC Irvine for grad school, where he did fundamental work with Cheryl Tsai on structure function of polyketide synthesis. He was a very productive grad student with 8 publications, including pubs in Nature and PNAS. So I was really lucky to recruit him to my lab, and he continued that productivity. So far, we have, I believe, 17 publications together, and more to come, I hope. Tyler really developed an entirely new area in my lab, namely cell-free biomanufacturing. When we started, I think most people thought it was crazy to think that you could put dozens of enzymes together in a pot and make them function within the kind of robustness required for commercial production. But as Tyler will describe, it actually is possible, and Tyler's really been the central player in making these systems work. Tyler's work convinced investors to launch our cell-free company, InDesign Technologies, where he is currently a director of research. And really, there's no way this company would have started without Tyler. At InDesign, Tyler's been able to now develop economical production of cannabinoids, and InDesign is now building a production facility to manufacturing diverse cannabinoids. So I think that's a great validation of Tyler's work and the cell-free approach in general. Unfortunately, since moving to the company, he has to keep some of his latest developments close to the vest. But I think the story he'll tell today will illustrate the power of the cell-free approach and why we're excited about it. So over to you, Tyler.
Okay, thank you, everybody. So yeah, today I'm going to tell you more of the background story, the technology development that went into founding InDesign, which was started actually the end of 2019. So kind of at an interesting time for everything. But throughout this, we've continued to grow. So the story I'll tell you today is about the development of cell-free systems for terpene and terpenoid production. So our overall arching goal when we first started this project was we wanted to develop an in vitro bioproduction platform that could take low-cost inputs like some kind of sugar and actually turn them into some useful compounds and build a system that really functioned continuously for long periods of time without intervention. With the idea that these breakdown modules could give you building blocks and energy that could then power a build module to make these useful compounds. And some of our initial targets that we wanted to focus on were especially in commodity chemicals, in particular terpenes for a variety of reasons. Terpenes are useful in a variety of different industries. And perhaps more importantly, they tend to be quite cytotoxic to cells, especially monoterpenes. So these were some of our first initial targets. And so the way that we went about this was actually quite simple. We still relied on metabolism as a blueprint. So this was nice for me for my background in primary and secondary metabolism, working in Cheryl's lab, where we just designed simply a series of enzymatic steps to take your input sugar and turn it into a product. And then we just threw away metabolism because we knew we weren't going to go back into a cell. And this gave us a lot of flexibility. At this point, we could swap enzymes for ones that were thermostable, had different cofactor preference, so on and so forth. And then instead of trying to reconstitute this whole pathway, on plasmids or in the genome, we just clone the individual enzymes in Max Express hosts like E. coli or yeast. We then isolate these enzymes. For all the work that I'm showing you, these were all nickel histag purified. But as we move forward, this can be as simple as just a heat treatment if your enzymes are sufficiently stable. You then have these isolated enzymes, you literally mix them together with your cofactors, add your substrate, and walk away. So that's the ideal. We're only adding the elements necessary for the transformation and regulation, and it's completely self-sufficient. So we think this has a lot of major advantages. You can reach really high yields. These systems are really easy to optimize because you have total control over units of activity that are input, cofactors, that sort of thing. The design build test cycles are really fast because you can, enzyme technology and expression cloning is really fast these days. So once you express and assay an enzyme, you can directly incorporate it. You have greater flexibility. We have some publications that I won't talk about where we've developed some non-natural cycles that wouldn't be possible in cells as well. And you don't have as much of a toxicity headache. At a certain level, when you're making so much product, you still denature proteins, which is a problem we have run into. But for all intents and purposes, it's much less than dealing with cells. There are definitely challenges with this approach. One of the common criticisms, people say, is enzyme cost. These enzymes are going to be expensive to produce. That's perhaps not entirely the case. And it also comes down to a question of enzyme stability. So if you have enzymes that are super stable and can last for long periods of time, it really will help address this enzyme cost issue. The bigger issue, and something that we were really aware of immediately, was cofactor recycling and maintenance, because CoA, NADPH, NADH, ATP, they're really expensive. So we didn't want to continually add these cofactors. We also didn't want to have to add a sacrificial substrate. So we wanted self-contained autoregulatory systems. And so, one of the limitations was, how do you develop these systems that can recycle their cofactors efficiently and have this whole process be autoregulatory? So this cell-free biocatalysis is not necessarily a new idea. People had reconstituted yeast glycolysis to make ethanol. But one of the main things that people did in order to address this cofactor issues, they developed stoichiometrically balanced systems, where if you input a sugar into a breakdown phase and you get NADH or ATP, you have to use those cofactors stoichiometrically in your build phase. The problem with that is that if you're cell-free, if you have any spontaneous hydrolysis or oxidation, the whole system will wind down and it won't last for very long. And so what we would ideally want is a system where you could build up excess energy in the form of these reducing equivalents or ATP, but still maintain carbon flux through the whole system. And so that was a key development, and one of the things that we worked on really early on to developing of these autoregulatory elements. So if we dig a little deeper into how this challenge presents itself, especially for systems that might not be, there might be a stoichiometric imbalance to begin with. So if we use glycolysis as our breakdown module, from 1.5 turns of glycolysis, you can get 3 acetyl-CoA as your 2 carbon building blocks, and you get some energy in the form of ATP and NADH. You can then use that to power the mevalonate pathway, which is shown here on the right. And it's nice because the acetyl-CoA, you use the acetyl-CoA and ATP stoichiometrically. So 1.5 turns gives you 3 acetyl-CoA, 3 ATP, and that's used perfectly by the mevalonate pathway. The problem arises when you turn to the reducing equivalent. So the mevalonate pathway only uses 2 NADPH, whereas glycolysis produces 6 reducing equivalents if you couple it through pyruvate dehydrogenase. So you make too much, which means you don't use enough, and it's of the wrong type. So this was a problem that we needed to reconcile before this would work at all. And so, this highlights kind of one of the issues moving cell-free is that you now, as you leave a cell, right, you remove all of those regulatory networks that are embedded within that cell to make life run. So we needed to develop regulatory networks that could just be dropped in that can account for this cofactor maintenance and imbalance problem. And so we've done that. We have numerous examples. What I'll talk to you today is about the Purge Valve, which we developed to regulate cofactor usage. We've also developed a similar system, albeit it's less enzymes, it's only 16, to regulate ATP, where you can use glycolysis to produce isobutanol. This actually works really, really well. We have a recent paper about that as well, if you're interested. So if we dig a little deeper into how the Purge Valve works, so if you take the simple biotransformation of pyruvate to acetyl-CoA using pyruvate dehydrogenase, this enzyme through this transformation actually makes NADH as a reducing equivalent. And if you add this enzyme NOX-E, NADH oxidase, this is a water-forming NADH oxidase, it basically reoxidazes NADH back to NAD. And in this process, you can just have carbon flux from pyruvate to acetyl-CoA without any buildup of reducing equivalents. If you are now able to develop a mutant pyruvate dehydrogenase that is specific for NADP over NADPH, you could then also produce the NADPH that you needed to drive a pathway, such as the mevalonate pathway, to make a terpene. It doesn't completely reconcile the balance, but when you have these three enzymes together, so the wild type PDH, the mutant PDH, and NOX-E, this is now an auto-regulatory system that allows you to balance the reducing equivalents. So when NADPH levels are low, when the system first starts, the flux is dominated through this mutant PDH. Over time, because you're not using enough of it to recycle all of it, eventually this side would shut down. However, because you have this wild type PDH where the reducing equivalents are constantly oxidized off, now flux dominates through the wild type node and flux is maintained. And so the key development here was kind of based off of previous work done with the E. coli E3 domain, where that enzyme had been re-engineered to have some specificity with an ADP. And we looked at the structure and overlaid the structure of a thermophilic PDH E3 and said, hey, we can make similar mutations. So we made a series of 4 different mutations in the NADPH binding kind of loop here, and showed that, okay, if the wild type has a preference for NAD over NADP, our mutant now almost has a perfect swap of cofactor specificity. So this was the missing piece that we needed in order to set up this full system and get it to run. And so this is an early paper, an early result demonstrating how the purge valve actually functions in an experiment. So this is a simplified version where you have the same conversion of pyruvate to acetyl-CoA using the purge valve system, but instead of coupling it to the mevalonate pathway, we're coupling it to polyhydroxybutyrate biosynthesis, which still has a stoichiometric imbalance. You make 2 acetyl-CoA, you only need 1 reducing equivalent. And so the way that this works, so we're monitoring NADPH levels here in the top graph at a variety of different ratios of the mutant PDH to the wild type. And you can monitor this at 340. And then polyhydroxybutyrate, it forms insoluble polymers, so you can monitor its formation by the turbidity. And so as the system starts, you see that the reducing equivalents quickly build up. But because you're not using them stoichiometrically, so as you're reducing acetyl-CoA into 3-hydroxybutyryl-CoA, you start to consume that NADPH, and then eventually it starts to build up again. However, once it starts to build up again, normally the system would shut down and you wouldn't see anything, which you can see by the low production here if you have too much of the wild type flux early on. But if you have a functioning purge valve, you now both have high reducing equivalents and you get quite a bit of product formation as well. This is also largely independent of the starting condition. So whether you start, with a tenfold differences in any reduction starting point, so NADP versus NADPH or NADH, it's largely independent. So you basically get the same conversion regardless of where you start, which was fantastic. So we also, in subsequent work, were able to show that this is a general concept. You could develop these purge valves almost at any reduction step within central metabolism. So as long as you could develop or have in hand an NAD-dependent reductase, an NADP-dependent reductase, and an NOX-E in hand as well. So these 2 reductases catalyze the same step. So for GAPDH, for instance, we developed a mutant GAPDH that was NADP specific as well, developed a purge valve there, and same for enzymes in the pentose phosphate pathway. So with all of these enzymes in hand, we wanted to now show that we could actually reconstitute this full enzyme cascade directly from glucose into a monoterpene. So this is at least a 23 enzyme, depending on some of the auxiliary enzymes that you added, step pathway in order to convert glucose into initially the monoterpene limonene. And so in this case, we used a purge valve at the GAPDH step instead of the PDH step, just due to simplicity, because PDH is a difficult complex to work with. And so when we evaluated this, we could evaluate the pieces because this is still cell-free, very easy. So we could do experiments where we left out various parts of the purge valve and we can monitor the effect on limonene production. And so if you leave out NOX-E, you actually don't get any or hardly any flux down to limonene. There's none produced. And if you use just one or the other of the gap DHs, probably due to the slight acceptance of both cofactors for each one, you do get some flux, although it's not great. But when you have the full system, the full purge valve, you get full conversion of limonene, fairly robust. This is within 24 hours. And so that was a great proof of concept. We have all the pieces in hand that work, and they work really well. And then we showed that some of our initial trials actually ran out of glucose. And so we started these with about 250 millimolar glucose, and we showed that these function for long periods of time. So at least in our case, 5 days, producing up to 10 grams per liter of limonene, which is well above the cell toxicity limit. And it's nice also cell-free because you have total control. You can also monitor what's going on with the cofactors as well. So we could see loss of inorganic phosphate through substrate level phosphorylation. And as the system winds down, build phosphate back up, and the ATP remains low because it's being recycled. We also showed that you could quickly make a variety of different monoterpenes simply by swapping out whatever terpene synthase you use. So really the only requirement was whether or not the terpene synthase expressed very well. And so we showed that you can make limonene, pinene, saponine. If you swapped out the farnesyl pyrophosphate synthase for one that makes farnesyl pyrophosphate, you could also make sesquiterpenes as well. So this worked really, really well. These are all at high levels, well above 10 grams per liter within about 4 or 5 days. So with that in hand, we knew that we could make large amounts of these isoprenyl pyrophosphate molecules. And so we turned our focus to kind of more commercial applications. And so one hot topic right now, and it's been for the past couple of years, is cannabinoid biosynthesis. And this is great because it funnels the terpene biosynthesis pathway that we had developed, but it also requires fatty acid biosynthesis, which is very complicated, as Thomas alluded to before. And so that would potentially raise a problem because the fatty acid biosynthetic pathway would also compete for the acetyl-CoA that's present. And within this transformation too, as I'll get to in a minute, the prenyltransferase is a membrane protein. So one of the first things we said, well, we can buy all talic acid, can we just add it directly? And that was the first thing. The other thing that we needed to do is develop a soluble prenyltransferase in order to do this biotransformation cell-free. And so we were lucky that within the literature, NPHB had been described by, I think it was Joel Noll's lab. And it actually does the conversion that we wanted of OA and GPP to CBGA. The issue is that it's very non-specific, and it actually prefers to make the C5 prenylated isomer instead of the one we wanted. So the idea was, could we use Rosetta design because there was a crystal structure of NPHB with GPP bound, could we then dock OA into the active site with the carbon in the correct position for acceptance of the prenyl group, and then vary these side chain residues in order to make that binding of OA better. It's an interesting story. We thought, initially we just designed an enzyme that was spectacularly inhibited by OA, but the results were quite different. So our initial round of 15 mutations that were output from Rosetta, 4 of those showed a significant increase in improvement, not just in rate of catalysis, but also in specificity. And when you combine some of those mutations, you now have NPHB variant that was very fast. So upwards of 1,000 to 3,000 fold improvement in the KCAT over KM. And that was absolutely specific for CBGA production. So it produced the correct stereoisomer. We also showed that you could do exactly what we had set out to, where you could use the GPP production pathway with the purge valve at GAPDH and feed OA exogenously from the beginning. And we could use this pathway to make GPP and then prenylate OA. And depending on which enzyme you use, so you could also make CBGA, you could also make CBGVA if you input diburonic acid instead of OA. So it was very flexible to the starting inputs as well. for these experiments, we were able to monitor periodic formation by HPLC and then confirm everything by NMR. So this was a great proof of concept. It's something that we continue to push the pedal on and really push the limits of what's possible with this pathway and others as well. With the idea that long term, can we develop highly robust sugar breakdown modules to make our building blocks and high energy cofactors, link them to build modules, but maybe not just one, but multiple different build modules to make different useful compounds. We have the flexibility to input alternative inputs, and then down the line, can we develop other sub-build modules to introduce other functionality as well? And so this is the team that all this work was done and pioneered in Jim's lab at UCLA. These are the names of the people that were involved in the work for the terpene work and that continue to be involved in some of our current grants from DOE, both on a BETO grant and RPE grant to further develop isobutanol production. Our InDesign team is growing quite well. We're at 10 right now. If you want to learn more or collaborate or join our team, so we are hiring. We just got a couple new grants as well and some existing ones. So yeah, feel free to reach out. And if you have any questions.
Great. Thanks so much for the talk, Tyler. It was really interesting work. I'm going to pass this to Anya, and she's going to moderate the questions.
Hi, Tyler. Thank you so much for your talk and the tour de force of thermodynamics and building complexity with biology. This is definitely a source of inspiration for what we do here. So I was curious if you could comment a little bit on, when you design those very complex pathways, how much time do you kind of need to design the pathway itself, and how much do you spend on enzyme discovery and then on putting this all together? Can you tell us a little bit about different steps in putting this cascade together?
Sure. So for the initial breakdown, glycolysis we knew was going to be ubiquitous, so that was almost an easy choice. Once we settled on glycolysis as a breakdown module, we quickly went out and just cloned a whole bunch of variants for every step. So we now have, and this was published recently in an isobutanol paper, we now have the whole system thermostabilized and everything's active as well. For the build phases, it's a little bit trickier. We pretty much stayed away from things that were complicated, like fatty acid biosynthesis, polyketo biosynthesis, that kind of thing. That's not to say that we haven't done work on that. So there was a follow-up paper to the cannabinoid work where we were able to make CoA compounds. So we were able to build OA cell-free as well. But that came later. We knew mevalonate pathway was relatively well-studied and ubiquitous. So we started kind of what was known. And then as we got good at using and replicating known technology, we then built it out further in terms of engineering stability for enzymes that there maybe weren't good analogs for, and then engineering specificity as needed. So, we let nature solve the problem where it could, and that's usually our starting point. But then knowing that we can always kind of lean back on our engineering skills to engineer specificity, stability, that kind of thing moving forward. It's turned out to be a great hybrid approach. And you don't have to worry about engineering the black box of the cell. So it makes that whole process easier as well.
Okay. Great, thanks. So we have quite a few questions in the Q&A. So I'm going to be allowing people to ask the questions live if they're okay with that. So Jonathan, would you like to ask your question, please? Jonathan Perkins. If not, I'm just gonna ask it.
Hi, sorry, can you hear me? I don't know if I can.
Yeah.
We hear you now, yeah.
Yeah, we can hear you now.
Great, I can't hear what anyone's saying, so my audio is super weird. I was just curious, like what form the proteins that you add individually take, like lysate, or do you actually like go through and purify the proteins completely?
So starting off and up until relatively recently, I'd say the past year or two, we were purifying everything using nickel and TA. as we started to develop more thermostable platforms, where we had every single step a thermostable variant, we've been slowly moving towards a more lysate-based system.
And I was also wondering, like, what's like the most complex, I guess, protein that you guys have been able to incorporate into these cell-free systems, or like,
So I mean,
things with like, I guess, not easily recyclable cofactors and things like that.
so the PDH is definitely that was a beast. So it's a tridomain protein. There's 3 different proteins or 4 depending on which organism you get it from. It has to be post-translationally modified. We were able to be relatively smart about how we produced it. So there was some data on the E. coli enzyme that you could express all the subunits together, and then just feed in lipoic acid, and it would do that lipoation. We were able to do a similar thing with the G. bacillus enzyme as well. So you could have a single basically expression and then heat treat to get active enzyme, which worked spectacularly instead of purifying everything together. There are some enzymes that we've started working with more recently that have different cofactors. Cofactor, you might think that that's a problem, things like FAD and some of these other things. As long as they're bound to the enzyme, they tend not to be. A lot of these are relatively stable. The NADH and ADPH are pretty stable actually in solution. And part of that is because we've been really smart about choosing thermostable enzymes that have significant activity at room temperature, so that temperature is not a concern to basically degrade the cofactors.
Cool. Thanks.
Great. So the next question will be from Cara Tracewell. Cara, we cannot hear you. If not, maybe Don. Don, would you like to ask your question live?
Sure. Very nice talk, Tyler. Thanks. What scale do you run the reactions on? What's the concentration of the enzymes typically? And how do you optimize the stoichiometry of the 22 different components?
Yeah, so we start at the mill scale. We have since expanded orders of magnitude larger than that. Especially for the acetbutyl, it works fantastic. So we're at hundreds of grams per liter as well. So in terms of, so that was one question. The other question was,
what is the concentration of the enzymes in the individual reactions?
Yeah, so we typically try to keep overall, so combined protein concentration less than 5 grams per liter.
For all of the components?
Combined, yeah, that's right. And then optimization is actually relatively straightforward. So because most of the steps you can couple to some type of readout like NADH production or oxidation, you can actually screen these as modules or submodules. So for like glycolysis, you can assay the top of glycolysis, you can assay the bottom of it, you can assay it all together if you're smart about it. And that's typically the approach we take. And then once you get into a system that actually functions, it's relatively easy to just optimize by kind of brute force to a certain extent, where you just add more or less and see how the effect on product formation. We've done some modeling as well, and it has informed, it more or less confirmed what our hypothesis was about bottlenecks, but it also helped us pinpoint what important cofactor levels might be. Like phosphate's an important thing for glycolysis, for instance, and units of some of the upstream enzymes as well, especially if they use ATP.
Okay, thanks.
So it's interesting to know that you studied modules rather than focusing on individual enzymes. Is it often that you have to study all or worry about all the KMs, KCATs of individual enzymes, or you still prefer to think about modules and only if absolutely necessary dive into individual kinetics?
So where we can, we don't worry about it. There are definitely some enzymes that have, and we have some issues with that now for some of the systems that we're developing at InDesign. Some of that you can get around by just adding more enzyme, which is nice, especially if it has a relatively high Km or low Kcat. That being said, it is something that we're aware of, and some of those enzymes you just have to add more of. And so these are things that you would design around, down the line when you can.
I see. And maybe last question for Janavi Prasad. Janavi, would you like to ask your question live?
Yeah, hi. So thanks. You already answered my question. But one other question I had was in many of these pathways, there's a lot of feedback inhibition and from downstream metabolites. So what do you do about that? So do you go about doing any allosteric engineering or?
So we usually, if we can, so the nice part is that there's usually so much literature out there that you can know upfront whether your enzyme may or may not be allosterically inhibited. And that being said, nature has probably found a solution to that problem. So for glycolysis, for instance, some of the phosphofructokinases are feedback inhibited. They're tightly regulated, that kind of thing. But you can usually find a variant that has a different type of allosteric regulation that will basically allow the whole system to be unregulated. And that's worked in a number of systems as well. So we've also done things with workarounds. So some of the work that I haven't published or presented here, it has been published. We got around the part of a dehydrogenase problem with basically engineering a bypass. So we could basically go from pyruvate to acetophosphate and then directly to acetyl-CoA. So there are also the potential for workarounds as well, depending on how you want to draw your schemes. So.